In this video I want to discuss Part B of Lab 23, and we're looking at two different features of alcohols, the pH and the solubility in water. And let me just talk about solubility in water first. So for alcohols, usually we say alcohols that have 1 to 4 carbons are going to be fairly soluble. Now, in some ways, just based on that, you could run through this list and sort of predict which one's going to be soluble or not. For example, cyclohexanol, there's 6 carbons. You might want to anticipate that that's going to be an insoluble alcohol. Now, all of these are liquids. The way we determine whether something is insoluble or soluble when two liquids are mixed together is that you're really looking for a cloudiness. It's a little bit different than the cloudiness that we've seen in precipitation reactions. What's happening here is that when you mix water and an insoluble liquid, like cyclohexanol, what you're going to see is an emulsion. And basically, there's like tiny little droplets of the insoluble liquid that gets suspended in the aqueous solution. It almost looks like vinegar and oil. Salad dressing when you shake it up and mix it up. And so in the video, I'll do that. I'll put the samples in a test tube, shake them up, and then briefly hold them in front of the camera for you to identify whether you see that emulsion or not. Let's go ahead and watch the video for the solubility. And the test tubes there, labeling from left to right. First one's ethanol. The second one is 2-propanol. Third one is 2-methyl-2-propanol. Fourth one, cyclohexanol. The fifth one, phenol. And the last one is the unknown. And then the second topic here in Part B is the pH of the solution. And what we're using here is a universal indicator. All right, that's what it's called. I'm doing a little bit different, I think, than the way the book describes it. Universal indicator is sort of like the red cabbage juice, where it's a single solution, and I'll put just a drop with each one of these samples, and then depending on what color it turns, then you can estimate what pH you think the sample's at. The image here is a little bit distorted, so just a little heads up about this. This is more of a green color, by the way. Notice that the basic pHs are not on this list because none of these are going to look basic. Okay, so I've narrowed down the color options for you. And when you record the information here, what I'd recommend doing is recording the color that you see and then the approximate pH that you think it is. And let me go ahead and discuss one particular alcohol. Let's look at the phenol group here. So phenol is our aromatic alcohol. And it does something that is unique, at least with respect to these particular alcohols and most alcohols. It actually undergoes a hydrolysis reaction like we saw back in Chapter 10 with acid-base chemistry. And what we see is that this is actually, that proton right there gets donated over to the water. And then this deprotonated alcohol looks like this, the negative charge. And then we have hydronium there. And so we could go through and then also call this a conjugate base, and then this a conjugate acid. None of the other alcohols do this kind of reaction. It also makes phenol more soluble than one would expect. Now, I'm not saying whether it's soluble or insoluble. I'll let you decide when you watch the video. But it does do something different. Okay? And it's all because of that aromatic ring there. This part is not going to be on the test, but that negative charge can actually be dispersed around that ring shared by all those carbons in there through resonance, something you probably learned in 151 at least a little bit. It stabilizes this product, and so what it does is it allows the reaction to proceed forward a little bit more than one might expect.